Hello and welcome to Chapter 12. Today we will be looking at how the brain gets organized and how the structures within each region and how regions communicate between each other. So during embryonic development, the brain grows faster than the surrounding membranous skull, so it must fold to occupy available space. It starts out as a neural tube. Okay, so it looks like this during development. And then as it grows, it gets swellings. And then this foremost area is called the telencephalon, and it starts to move forward and then it starts to move back. And while it does this, it makes little ridges, and that will increase surface area. In this class, we study the adult brain. So you do not need to know the telencephalon, the mesencephalon. You will need to know the diencephalon because it's still part of the adult structure. What you will need to understand is this, the domains of the cerebral cortex. We'll go into that in great detail. And then the structures that make up the diencephalon. So that would be the thalamus, the hypothalamus, and the epithalamus, as well as characteristics of those structures. You will need to know what three structures make up the brainstem: the midbrain, the pons, the medulla oblongata, as well as characteristics of those three structures. Then if we move down, this will be the spinal cord, beginning a separate region of the central nervous system. And then this outgrowth developed into the cerebellum, which we will talk about next week. Adult brains have four regions: the cerebral hemisphere. So when you think of what a brain looks like in that folded structure, those are the cerebral hemispheres. So we have a right cerebral hemisphere and a left cerebral hemisphere. And within those cerebral hemispheres, it will be divided up into lobes. The diencephalon, as I mentioned, is made up of the thalamus, the hypothalamus, and the epithalamus. The brainstem is made up of the midbrain, the pons, and the medulla oblongata. And then we'll talk about the cerebellum next week. These convoluted structures, which is what most of us think about when we think about a brain, these are the cerebral hemispheres. There's a left one and a right one. The diencephalon, that thalamus, hypothalamus, and epithalamus, it forms like an egg-like structure, kind of sitting in the very inner deep section of your brain. Coming off of that diencephalon, we then have the brain stem, which these three regions have very distinctive functions from each other. We have the midbrain, which has what are called nuclei, islands of cell bodies. This is the pons, and these are going to be mainly conducting tracts that connect different parts of the brain or different parts of the brain to the cerebellum. And then we have the medulla oblongata, which is going to control a lot of basal functions. As we mentioned in lab last week, if you look at the spinal cord, you have gray structures that are deep, and then it's surrounded by white matter with a central canal in the middle. Well, these cell bodies are going to send axons, what are called through ascending tracks. So tracks of axons, bundles of axons, that are going to go from the spinal cord up into the brain. So these are ascending tracks. These are axons. And so the axons are then going to run into different brain structures, and then they will be acted upon by the cell bodies. And so while we have these axon, these white fiber tracks that are going to lead up to the brain tissue. And so this will be the deep section, and then it'll be surrounded by gray matter. So you do need to know the differences and patterns between gray matter and white matter, between the spinal cord and the brain. Gray matter are things that are not myelinated. So neuron cell bodies and dendrites, and it says short non-myelinated neurons. So axons can be myelinated or not myelinated. If they're really small, they might not be. But mainly it's neuron cell bodies and dendrites. White matter is made up mostly of myelinated axons. You can have some small non-myelinated axons in there as well, but the white color comes from the myelin. So those are going to be myelinated axons, giving it that very white appearance. As I mentioned, so as you go up from the spinal cord, there are ascending tracks, and then there are descending tracks. The ascending tracks or the descending tracks come up through this white matter. These are myelinated axons, and they can interface with this gray matter made up of cell bodies and dendrites. This is in the cerebrum. The cerebellum has a similar structure, minus the islands of white matter on the interior with a peripheral layer of gray. This is why we have to be careful with concussions, because the cell bodies of the brain, so those neurons, are located just right underneath your skull and can be damaged pretty easily. These are called islands or nuclei. They are made up of clusters of cell bodies, and they each have unique functions. Ventricles are fluid-filled chambers that are continuous to one another and to the central canal of the spinal cord. The brain requires a lot of glucose. It needs a very clean environment, so the waste will be taken away. It also requires a unique ion concentration. So the brain and the spinal cord get bathed in their own special fluid, and that is cerebrospinal fluid, and it will be produced in the ventricles and then circulated by ependymal cells, a specialized type of neuroglial cell. There are 4 ventricles, and you do need to know where these 4 ventricles are located. The first 2 are called lateral ventricles. So lateral 1 and lateral 2. In lab, we don't ask you to, because we'll be talking about ventricles in lab as well, and you won't need to know which one is on the right, which one's on the left, just that these are lateral ventricles. This blue shading is showing the cerebrospinal fluid volume located in each of those ventricles. So the lateral ventricles are located just under the corpus callosum, and we'll be talking about that structure as well. And then in between, so we have one in the right hemisphere and one in the left. In between, we have something called a third ventricle. And that third ventricle is going to surround the thalamus. And this little hole here is something called a thalamic adhesion, which the thalamus is like 2 halves of an Easter egg that's been put together. And that adhesion is like a rivet that keeps those 2 masses together and kind of operating as one unit. Then leading from the third ventricle down to the fourth ventricle is the cerebral aqueduct, and it looks like a stem. We'll look at a ventricle model in lab. It looks like a stem. The fourth ventricle is rather large. It is going to be at the opening to the cerebellum. So you do need to know that there are 4 ventricles plus the cerebral aqueduct and what structures they are located next to. So the lateral ventricle is underneath the corpus callosum. The third ventricle is surrounding the thalamus. Then we have the cerebral aqueduct between the third and the fourth. And the fourth ventricle is going to be located at the opening to the cerebellum. There's also a membrane in between the lateral ventricles called the septum pellucidum. Okay, so now we're going to look at the cerebral hemispheres. So the cerebrum, it is the superior part of the brain. It's the part that's all folded. If you were to put your finger on the top of a brain, you would be feeling a gyrus. And then because the brain tissue is folded, we have shallow grooves called a sulcus, and we have extremely deep grooves called a fissure. And a fissure is going to be separating 2 major structures, whereas you'll have a lot of sulcuses all throughout the brain. Okay. So there are 2 hemispheres, a right and a left. And you need to know 2 fissures and 2 sulcuses. So the fissures that you need to know are the longitudinal fissures. So it runs down here, it separates it into a right and a left hemisphere. And then the transverse fissure is in the very back, and it separates the cerebrum and the cerebellum. So the sulcus that you will need to know is called the central sulcus. It's going to divide the frontal lobe and the parietal lobe. And it's also very important. It's structural, but it's also very important functionally because it divides the motor areas from the sensory areas. That's why we make you learn it. Each hemisphere is divided into 5 lobes, and it really depends on the textbook that you have. Some textbooks will say that there are 4 lobes, and some textbooks will say that there are 5. And the reason why this is this 5th lobe, the insular lobe, is located very deep, and it contains the deepest portions of the frontal, parietal, and temporal lobes. So some textbooks will just give you 4 lobes, and then some textbooks like ours will give you a 5th lobe, which is the deepest part of those structures of the lobes. Okay, so we have a frontal lobe, and it is named for the frontal skull bone. We have parietal lobes, have a right and left temporal lobe, and we have an occipital lobe. Okay. Each hemisphere, so right and left, have a cerebral cortex. That is that outermost layer of gray matter, then white matter, and basal nuclei. So that's from here, where we have each lobe has gray matter on the outermost surface, white matter deep, and then these basal nuclei, which are clusters of cell bodies located within the cerebrum. Okay, so now we're going to talk about the cerebral cortex. So the cerebral cortex is the most superficial part of the cerebrum. It is only 2 to 4 millimeters thick. It is heavily folded, and it has a superficial layer of gray matter. It is the site of our conscious mind, of awareness, our personality, sensory perception, voluntary motor initiation, communication, memory, storage, and understanding. All cortex neurons are interneurons. So they send and receive information to other neurons. So that's why this is our thinking brain. It's going to receive information from other neurons. It will process that information. It will send information on to other neurons. So it's that integration area, and they're all what are called interneurons. We have billions of neurons. They are arranged in 6 layers. You do not need to know which neuron types are within each, just to know that there are different layers, different levels, and different types of neurons found in the cerebral cortex. There are 3 main types of functional areas. Motor areas control voluntary, they control skeletal muscle movement. Okay. This is not going to be the area that controls the autonomic nervous system. So these motor areas control voluntary movement. Sensor areas are going to get input from the environment, input from your internal environment, sensory areas, proprioceptors, and bring that information to your brain. And then there are association areas that require either different lobes or different parts of a lobe, or they're just more complicated. Each hemisphere is concerned with contralateral control, opposite side of the body control. And this is because there's something called crossing over that occurs at the medulla oblongata, and we'll get to that. But our cerebral cortex, is going to be involved with contralateral control. So we're going to be moving on to domains, and there we need to cover 14 of them, and these are discrete cortical areas. So they are located in one lobe, and they will control a specific motor or sensory function. So domains are a single lobe and pretty well-defined functions. We'll have things called multimodal areas for higher functions. These are going to be spread over many areas. Okay. So we're going to start a look at our 14 domains, and each one is located in a single lobe. So you need to be able to identify which lobe and what is its function. First, is it motor? Is it sensory? And then what function, what purpose does it do? We are going to start off with looking at the main motor areas. So the main motor areas are all located in the frontal lobe. So we have 4 domains located in the frontal lobe: the primary motor cortex, the premotor cortex, Broca's area, and the frontal eye field. 14 domains sounds like a lot of domains, but you'll start to notice a pattern. Okay, so each lobe usually has a primary, and then for the frontal lobe, it'll be premotor, but for other lobes, it will be association. So we'll start with the primary motor cortex. The primary motor cortex is part of the frontal lobe. It is made up of large pyramidal neurons. So if you remember when we were looking at that table of different kinds of neurons, we have Purkinje neurons, which we'll talk about when we get to the cerebellum. And for now, we're going to be talking about pyramidal neurons. So pyramidal neurons are kind of like the Hulk of neurons. They are large, they have large axons, they are going to project all the way down to the spinal cord. So these are massive neurons, and they are going to control conscious, precise, skilled skeletal muscle movement. So if you are playing the piano, then your primary motor cortex is sending those messages down to the muscles of your fingers and telling them when to contract or when to relax. So pyramidal neurons of the primary motor cortex are going to send the message to have structures physically contract. Pyramidal tracts, these are bundles of axons, are going to project all the way down the spinal cord. Next to the primary motor cortex, we have the premotor cortex. So it's not helpful if you want to play a trumpet or a piano to contract random muscles in your fingers. It probably wouldn't make beautiful music. So the premotor cortex is the area of the cerebrum that plans movements for skilled motor activities. It will be responsible for the amount of force, for planning the timing between movements, planning which muscles will contract at the same time or in sequence. So the premotor cortex is the planner, the organizer. It controls learned, repetitious or patterned motor skills. It sends that input to the motor cortex, or it does have access to a small number of motor neurons. However, for the exam sake, if we're talking about voluntary muscle movement, the actual sending message to muscles to contract, that is the primary motor cortex. For the exam for premotor, we are focusing on planning. So one of the kind of categories of questions on your exam is, if you damage the premotor cortex, you would not be able to do which of the following? Or you would be able to do something. What would you still be able to do? So if you damaged the premotor cortex, you would unlearn or lose learned behavior, but you would be able to learn it again. So if you previously knew how to play Twinkle, Twinkle, Little Star, and you damaged the premotor cortex that held that movement pattern, with training, you would still be able to play Twinkle, Twinkle, Little Star. If you damaged the motor cortex, you would lose the physical ability to contract those muscles. So you may be able to think it in your head, but you would not actually be able to press those piano keys. Okay. So do you know how damage to a cortex, to a cortical domain, would affect what a person could or could not do? Okay, so we're already down 2 domains, on to numbers 3 and 4. We have Broca's area and the frontal eye field. So Broca's area is present in only one hemisphere. We usually have most things present in both hemispheres, but they're not duplicate. But Broca's is present in only one hemisphere, usually present in the left hemisphere. And because of that contralateral control, if Broca's area is present in the left hemisphere, people are right-handed. That's about 90% of the population. The other population, Broca's area is in the right side, and so they are left-handed. It is located in the frontal lobe because it controls the muscles for speech production. Broca's also has kind of a little association area or a premotor area of its own where it's involved in planning speech and other motor activities associated with speech and language. So, but we do pretty much focus on the motor speech function, the muscles of speech, because we're trying to focus on the frontal lobe being in control of motor activities. The frontal eye field is going to control voluntary eye movements. So this is a map of those areas. If you look in, so this is the central sulcus. It divides the frontal lobe from the parietal lobe. The primary motor cortex is just anterior to that central sulcus. And then adjacent to the primary motor cortex is the premotor cortex that does the planning for the primary motor cortex. Then we have the frontal eye field is even more anterior. And then in one hemisphere, we're going to have Broca's area. So this is a somewhat interesting view of how we can interpret our body. But this is showing that motor neurons are going to be grouped together for similar areas. And it just makes things more efficient. If we want to move our fingers, then we would be looking at motor neurons located in this area. If we wanted to, so if you can see the face is disproportionately represented. So more neurons are given to things like speech and facial expression than there are to like the shoulder, arm, and elbow. This map is similar when we're looking at sensory. We have a disproportionate amount of neurons that are going to be involved with sensory information. And then in our fingers, as compared to things like our arm and elbow and hip and leg. You don't need to memorize this in any way. You do need to understand that like neurons are grouped together in areas of that cerebrum, and that the number of neurons is not proportionate to like body area. It is actually very disproportionate to body area with much greater representation of our face and hands. Okay, so now we're going to move from frontal lobe and motor to parietal lobes and sensory. So just on this other side of the central sulcus, so here we have our primary motor cortex on the frontal lobe, our central sulcus, and then we're going to have our primary somatosensory cortex. Okay, so we're going to look at these areas next, and they are color coordinated with their lobe. So we're going to move from front to back, and the rest, these are all sensory. So frontal lobe is motor and everything else is sensory. Okay. So if we're looking at the parietal lobe, just on the other side of the central sulcus is the primary somatosensory cortex. It is going to receive sensory information from the skin and from proprioceptors that are found in skeletal muscle, joints, and tendons. The primary somatosensory cortex is also going to be the area that tells your brain where that information is coming from. So if I get a splinter in my finger, then the primary somatosensory cortex will say, oh, that splinter, that pain is coming from her left pinky. And it'll be way more specific than that, but it will tell what side of the body and what portion of the body. And that ability is called spatial discrimination. There are also things called discrimination tests that we can do on skin that can test for damage of the primary somatosensory cortex. But its primary purpose, so it's located in the parietal lobe, and it has two functions: to receive general sensory information from skin and proprioceptors, and to tell where that sensory information is coming from. And that ability is called spatial discrimination. So moving on to domain number 6, we have the somatosensory association cortex. So we may know that we have pain or pressure or light touch or pressure sensors are getting activated, and we might know where they're getting activated. But it's the responsibility of the somatosensory association cortex to take that sensory information and to put understanding to it. So if you put your hand in your backpack, you can discriminate between a pencil or a calculator or your phone or your earbuds, and that is because of the somatosensory association cortex. It determines size, texture, and the relationship of parts of objects being felt. So you might feel the buttons on your calculator, and that's how you determine it's your calculator and not your phone. It grows as we grow. So these are learned. We learn better association as we get older. Okay, so primary somatosensory gets the raw sensory information. It's hot, it's cold, there's pressure, where's that pressure coming from? And then it's the somatosensory association cortex that makes sense of that information. So if you damaged your primary somatosensory cortex, you would lose the ability to get sensory, to receive sensory information from different areas of your body, depending on where the damage occurs. So the sensory endings in your fingers are still sending that information, but it's not received. So that phone is never picked up and says, yeah, I hear you, you know, that hurts, or it's hot, it's cold, there's pressure. The phone just stays off. Somatosensory association cortex, if it is damaged, you can actually still feel something, but you don't know what it is. So if a part of a somatosensory association cortex was damaged, you could lose the ability to know if something is like a pillow or is it a book. You would lose the ability to determine, like if you stick your hand in a bag, what is in that bag. Is it a coin? You lose that ability to associate what you're feeling. Okay. So now we're working our way back to the occipital lobe. So as we'll discuss a little bit more, especially in lab, you have the eyes, and they are going to send out optic nerves, which become tracts, and they cross over at the optic chiasma. And so they have optic nerves to the optic chiasma and then optic tracts. And we'll discuss that in much greater detail in lab. But those axon tracts go through your brain to the occipital lobe. And the primary visual cortex is going to receive information from the retina. So it will say, I have a black spot here, and I have a black spot here, and then I have a few orange spots, And then I have some more black spots. And this is what I have. So those photoreceptors are stimulated and the visual cortex receives that information. Well, I'm hoping that you've guessed maybe what I've attempted to draw. And your next domain is your visual association area. And so it takes this raw information, and we actually don't receive a ton of information. We don't receive like solid structures. We receive parts of structures, and our brain fills the rest of it in. So maybe you're guessing that I drew a snowman. At least that's what I was trying to draw. Some people might say a smiley face. And the reason why people give different answers is because this is a very learned area. We use past visual experiences to interpret visual stimuli. It's complex processing involving the entire posterior half of the cerebral hemisphere. But the key part of this is that it's learned. So it's why witness recollection is so variable between people. Because if you grew up in, like Montana, and you saw somebody and they were wearing a red hat, you might say, oh, well, that person, of course, was wearing like a snow hat with like the little fuzzy thing off of it. But maybe someone who grew up in Florida is not used to skiing. You're like, no, that wasn't that. It was a red baseball cap. And you get someone from Ohio and they say, that wasn't just a red baseball cap, that was an Ohio State baseball cap. So where a person grows up or how a person grows up, and especially when a person grew up, like an older person versus a younger person, they are going to have and honestly see different things because their brain has to fill this image in with what they know. So as we get older, we think we know more. So someone who's older, who sees someone running and has long hair, they might say, oh, that person was a girl. Whereas somebody who's younger and hipper might say, oh no, that wasn't a girl, that was a guy. He just happened to have long hair. So a person interprets data and they're honestly seeing something different in their brain. Okay, so anyway, back to our domains. So we have two for the occipital lobe. Primary visual cortex, that is going to receive information from the photoreceptors via the optic tracts. And then the visual association area is going to take that primary visual information and translate it into an image. So moving on to the temporal lobes, we have a primary auditory cortex, okay, just like we had a primary visual cortex and a primary somatosensory cortex and a primary motor cortex. So you can kind of start to see there's this pattern. In the temporal lobe, we have the primary auditory cortex, and it is going to receive auditory information. And the way that your ears are designed, they have hair cells that get bent over, and it's in the cochlea, and the further along it goes, the higher the pitch. So the raw information that gets sent with hearing includes pitch and loudness, because loudness is how much is that hair cell bent over. That's why really loud noises can bend them over so much that they break. And they're not replaced, by the way. So be careful with your hearing. And then pitch is how far along it is in that cochlea. So the primary auditory cortex gets that auditory information. The auditory association area takes that raw data and says, oh, I know that song, or that sounds like jazz, or that sounds, like rock and roll, or it sounds like Adele, or it puts meaning to that information. So if you damage your primary auditory cortex, those hair cells still get bent over, it still sends those impulses, but again, it's not received. And then the auditory association area is learned. So if you damage the auditory association area, and right after you receive that damage, you could hear a song and you wouldn't recognize it, but you could relearn it. You could get a whole new appreciation for different kinds of music as you learn different forms of music. Okay, so the temporal lobe has one more primary olfactory cortex, and it is associated with the sense of smell. So we have olfactory nervelets that hang down into our nasal cavity, and it will take that information and send it to the primary olfactory cortex and make us aware of odors. And it will work with the limbic system to associate those odors as being good or bad. Okay. We have 3 more. And then for these 3, they are part of the insular lobe, and so they're much harder to access, and so we don't know as much about them. So for these 3, you do need to know that they're part of the insular lobe. And then for vestibular, it's involved in maintaining balance. Gustatory is involved in perceiving taste. And the visceral sensory area receives sensory information from visceral organs such as your lungs. If you feel like you need to breathe, that horrible feeling, that's like you've been underwater too long, that goes to the visceral sensory area. An upset stomach, a full bladder, those are all going to get relayed to the visceral sensory area for evaluation. So again, you need to know what lobe they're located in. We have the 4 motor areas of the frontal lobe, and then we have 10 more associated with the primary lobe and the temporal lobe, the occipital lobe, and then the insular lobe. So some information is too complex to be assigned to a single area. So we assign those, we call those multimodal association areas. So these usually involve multiple lobes and multiple areas. And the flow of information still will be involved with a sensory, like the primary sensory cortex to sensory association cortex, and then it will get further evaluation by this multimodal association cortex. And then the multimodal association cortex can send output to multiple areas in the brain. This is going to this allows us to give meaning to information received, store information in memory. That is really interesting. We will talk about that more in a little bit. And tie information to previous experience and decide on actions. We store memories throughout our brain, but we store it in the lobe that has a meaningful association area. So if we went to a carnival and it was filled with bright, colorful costumes, and what we remember most is what it looked like, we would store that memory in the occipital lobe. If we went to your first rock concert and you remember the music and it's probably really loud, then you're going to store that memory in the temporal lobe. So whatever is the dominant sense, that's where that memory is stored. If you don't access memories, so the way we maintain a memory is we change its plasma membrane and kind of fix it. But if we don't access memories at all, then it weakens and we can lose that memory. So the strongest memories are those that are accessed most frequently. And with so many pictures that we have now, it's a lot easier to stabilize those memories and to keep actually the memory healthy. That makes sense. Sensations, thoughts, emotions become conscious and makes us who we are. So multimodal association areas are what we think about when we think about being human. Okay. They are broadly divided into 3 parts: the anterior association area, the posterior association area, and the limbic association area. The anterior association area is also called our prefrontal cortex. It is the most complicated cortical region. It's involved with intellect, cognition, recall, and personality. It contains our working memory needed for abstract ideas, judgment, reasoning, persistence, and planning. So our anterior association area, our prefrontal cortex, contains the area that we think of when we think of being human. If it were to be damaged, we could have a different personality. It also is so important because of what's called working memory, which is the idea, it's the ability to hold one piece of information in your brain and add another piece of information and then relate them to one another. So it can also be in cognitive, like the ability to hold, okay, I need a subject and a verb and like an object, and they need to agree. So that's multiple pieces being held together at once. And without the ability to have a working memory, everything else is so much harder. Language and expression. And working memory is what we think of as higher thinking. We need a good working memory for higher thinking. Planning, thinking, oh, wait, I can't do that on Monday because I already have something else planned. Or long-term planning, oh, okay, so I'm getting a new apartment in 6 months. What do I need to do in order to be ready to move in? I need to turn on my utilities. I need to maybe buy a bed. I need to get internet. So all of those skills are part of the prefrontal cortex. Your prefrontal cortex isn't complete until the age of somewhere, they used to say 18, and now the new estimates is 24. It's highly dependent on feedback from the social environment. So if you're in a safe, healthy environment with lots of abilities to use your working memory or express your personality, then it will grow and form lots of connections. The posterior association area, so that's located in the temporal, parietal, and occipital lobes, plays a role in recognizing patterns and faces and localizing us in space. So if you walk outside your house and you're going to go to your friend's house or the library or the post office, and you go, oh yeah, I think it's this way, I think it's pretty far, or I think it's really close, then that is the posterior association area. Some of us have a better one than others, and it's kind of that sense of direction. It's also the area that tells us if we're up 5 stories or if we're on the basement level. So that localizing in space is north, south, east, and west, but it's also elevation. You know, are we higher or lower? Kind of how high are we up there? It's also important in recognizing patterns, like it could recognize a face, it could recognize a gait, just so that's kind of interesting. And then a new area that hasn't been known of really for, I mean, maybe 5-10 years, which is pretty new, is that there is a second area for written and spoken language located in that occipital lobe, which I just mentioned because it's an interesting area of research. But for language, really think of Broca's area. For posterior association area, keep focused on recognizing patterns and localizing us in our general area, or knowing where to go if we're trying to get somewhere. Limbic association area is part of the limbic system, and it is involved in emotions, and it is located pretty deep inside your brain. It does play a role in establishing memories, but that's also somewhat neurotransmitter related. But for now, when you think of limbic area, think of emotions. So as I mentioned earlier, we have similar but not identical functions in the right and the left hemisphere. So even if we're looking at like our primary auditory cortex on the right side and the left side, they're going to be getting input from the right ear and the left ear. They have similar jobs, but they're not identical. You can't lose one and have the same function. So you can't lose the primary auditory cortex on the right side and have the same hearing, and they're not redundant. So they're similar, but they're not redundant. Cerebral dominance refers to the hemisphere that is dominant for language. So where is Broca's area? 90% of humans have Broca's on their left cerebral hemisphere, and they are going to be usually right-handed. And that left side of the cerebral hemisphere tends to be more, contain more connections for language, math, and logic. So the other 10% have Broca's in their right side. These tend to be your more artistic or your more musically inclined individuals, and they do tend to be left-handed. Okay, and establishing hand dominance is actually a very important developmental milestone for kids. Hemispheres communicate almost instantaneously via fiber tracks and functional integration. Okay, so now back to the three basic regions of the cerebral hemisphere. We've talked about those 14 domains and those 3 multimodal association areas of the cortex. So we're going to move on to white matter and basal nuclei. But first, a few definitions. Ganglia are a collection of neuron cell bodies in the peripheral nervous system. Nuclei are clusters of densely packed neuron cell bodies in the central nervous system. Nerves are collections of axons In the peripheral nervous system, tracks are bundles of axons in the central nervous system. For cerebral white matter, so we've discussed the little gray folded areas, those 2 to 4 millimeters on the very exterior part of the surface. And now we're going to talk about the white matter. So white matter is made up of myelinated fibers bundled into large tracts. They are classified according to the direction they run. There are 3 kinds: association, commissural, and projection fibers. Association fibers stay within the same hemisphere. They can be very short between gyri or long between lobes. But we're still going to be looking at like the right parietal lobe to the right temporal lobe, or the right portion, on the right cerebral hemisphere of the frontal to the parietal. So we're looking within the same hemisphere. Commissural fibers occur between the right and left hemisphere. The largest one is the corpus callosum. There's also one called the fornix. And these are going to help the right and left hemisphere communicate. We also have projection fibers. Projection fibers are going to connect the hemispheres with the lower brain or spinal cord. They have a much greater range, and then they are also going to cross over at the medulla oblongata. Because they cross over at the medulla oblongata, we have contralateral control. So the right hemisphere is going to control the left side of the brain. The left hemisphere controls the right side of the brain. And that's due to crossing over at the medulla oblongata. So this picture shows association fibers. They're going to stay within the left hemisphere or within the right hemisphere, and they can will just go from one area or one lobe to another within the same hemisphere. Commissural fibers, such as the corpus callosum, shown here, are going to connect the right and the left hemisphere. And then we have projection fibers. These are coming from the frontal lobe but they can come from elsewhere. These are actually coming from the parietal lobe too. So they will come from the cerebral cortex, and then they will come down here to the medulla oblongata, where it's called decussation, crossing over of the pyramids. Pyramids meaning pyramidal neurons. So the pyramidal neurons are crossing over at the medulla oblongata, ending up in contralateral control. Okay, so here is the cortex, and here is the white matter, and then we have these islands of nuclei called basal nuclei. Our basal nuclei are the third region of the cerebral hemispheres. Their function is to influence movement. So they're going to work with the premotor and prefrontal cortex. So if you look at where the basal nuclei are, you have these fibers that pass through them. So those basal nuclei are going to start and stop movements, monitor velocity, and inhibit antagonist unnecessary movements. They also play a role in cognition and emotion to filter out correct or inappropriate responses. So if you are thinking of saying something to a classmate or to your friend, and your mom walks in, your basal nuclei might stop what you're saying because you're like, oh, I can't say that with that person there. So they will play a role in editing movement or editing a behavior. And Parkinson's and Huntington's disease are disorders of the basal nuclei, but those are neurotransmitter related. Okay, on to our last section of tonight. We're going to talk about the diencephalon. The diencephalon is made up of 3 gray matter structures. So the thalamus and the hypothalamus are what are called clusters of nuclei. So the thalamus is very round, and then the hypothalamus is a little bit different shaped. But inside each of them, if you think of this as like an Easter egg, it's like putting a bunch of jelly beans inside. And each of those jelly beans is a cluster of cell bodies that is going to perform a specific function for either the thalamus or the hypothalamus. For the epithalamus, there's part of it that becomes the retina, and that part we're really going to look at is the pineal gland. So you do need to know the 3 structures that make up the diencephalon and what their functions are. The thalamus is the most centrally located of those 3 structures. And it is considered the gateway to the cerebral cortex. It is a relay station. It is a relay station for all sensory information coming into the cortex. So if there is sensory information coming from your skin, it all has to pass through the thalamus so that it gets sent to the right place on that primary somatosensory cortex. So we have, and it's going to sort and edit information before sending it on. So if it's getting several inputs that need to go, that are coming from like your right leg, so it needs to go to that part of the primary somatosensory cortex, it's going to group that and send it as a single packet of information. Sometimes it will edit, actually like 99% of information it receives, it edits out. It just doesn't pass it along. It doesn't reach the threshold, the importance level, the frequency of stimulation to get passed along to the cerebral cortex for evaluation. If it didn't edit out 99%, we would just be overwhelmed by the amount of sensory input. It can also take sensory information from areas such as the hypothalamus and the cerebellum and basal nuclei and send it elsewhere in the body, in the brain as well. Okay, so it sorts, edits, groups, and relays ascending input. All sensory afferent nerve impulses have to go through the thalamus. It also gets input from the hypothalamus and the cerebellum. We'll talk about the cerebellum next week. Overall, it acts to mediate sensation and motor activities, regulate emotion, learning, and memory. The big thing is really that it sorts, edits, and relays ascending input. So we have the thalamus, and then directly underneath the thalamus, we have the hypothalamus. So the hypothalamus is extremely important, and we'll talk about it when we get to the endocrine system, and we're going to talk about it now. So we're going to talk a little bit about different functions then and now. For now, we're going to look at how it is involved in controlling homeostasis. So we haven't talked about the blood-brain barrier, we'll get there. The hypothalamus, the blood-brain barrier is absent. So it has a lot of homeostatic sensors. So it has glucose sensors, it has osmosensor, it has baroreceptors, It has, so it can measure oxygen levels, just all sorts of chemoreceptors. It also attaches to the pituitary gland, but that's really something that we're going to look at later when we get to the endocrine system. So the hypothalamus is the main homeostatic regulator. It controls the autonomic nervous system. It has input into the autonomic nervous system for baroreceptors, which would affect blood pressure, heart rate, pupil size. We'll go into that when we talk about cranial nerves in a little bit. Initiates physical responses to emotions. So the neurons that run from the limbic system, they actually run through the hypothalamus. And so when your limbic system is highly stimulated, something's really upsetting you, it could be good, it could be bad, those axons and those neurons are so leaky that they start to affect the sensors in the hypothalamus. And so this is why if you're really stressed, like, and it could be good, it could be bad. I think I lost like 20 pounds in like the 2 months leading up to my wedding, which I was super excited about, but super stressed out about too. But because those axons run through the hypothalamus, it'll change your hunger and satiety. It also has thermoreceptors to help regulate body temperature. So it's very involved in maintaining homeostasis. It also regulates water balance and thirst through osmoreceptors. It's part of the sleep-wake cycle, and then will function on the endocrine system and its role in the endocrine system when we get to the endocrine system. So don't worry about the endocrine system functions. Do know the homeostasis ones: body temperature, hunger, satiety, autonomic nervous system, osmoreceptors, water balance, thirst. The epithalamus is the third component of the diencephalon, and the structure that we're going to look at is the pineal gland. It is very deep in your brain, and it secretes melatonin that helps regulate the sleep-wake cycle. We'll talk about other functions of melatonin when we get to the endocrine system. And that's why, in a way, you need to be careful how much you take, because melatonin is a hormone. And yes, it does help regulate sleep-wake, but as I will tell you later, it helps to regulate other things too. Okay, so that is the end of our lecture today, and we'll pick up with section 12.4, lecture 12B next Tuesday. Okay, have a great weekend.